and sausage and gravy. Yes. Omelets. All sounds yummy to me. I'm trying to make myself. Ah, there we are. You hear that? That would be a snow plow right outside the window. Let's see if I can hold my lighting here. Now I look like extra ghost-like. Man, this winter weather. This lighting is not helping me. Awesome. Somebody got a pep talk yesterday. Eggs, fried potatoes, and sausage. Yes. That sounds amazing. All these things sound amazing. I love breakfast food. Oh, maybe not goat's milk with spiderweb. I'm not sure I'm down with that. You know what? I'm not big on cereal either. I like a good hot breakfast. I don't dislike cereal. I just like warm. Even oatmeal with lots of cinnamon, brown sugar, blueberries. That's a good simple one. All right. Well, yeah, sometimes. Like if I'm not making myself a real big breakfast, like I'd rather have. Sweet deal. Well, today we are going to stomp all over some goals. Okay. We are going to look today at, and I'm going to just do these for the sake of quickness because I want to go through this. I want to take a quiz and then I want to go through a little bit more. Or I might go through it all and then have you quiz. That's what we'll do. Okay. But we are a couple quizzes behind, and then that will make us behind on our unit test, and nobody wants to do that. Ooh, waffles. Delicious. Well, I also like peanut butter on my waffles. All right. So what is recombinant DNA? We're going to learn about what recombinant DNA is. We know what DNA is, right? Ready to give me a green check if you know what DNA is. Hopefully we aren't already here and don't know what DNA is. So we're going to take DNA. What is recombinant DNA? We're also going to look at why would a scientist insert DNA of one organism into the DNA of another organism? Why would we do that? And then we're going to take it up the triangle today to our analysis section and talk about what are some pros and cons of genetic engineering and look at what in the world, Nutella, is genetic engineering in general. Good. We are familiar with DNA. So let's look at this picture. Biotechnology is the science that involves using living organisms to produce needed materials. All right. So using an organism to benefit another organism. So we're going to use life, right? That's what this is. Bio is life technology. So what is biotechnology? Well, it is living technology. How we can use living things, yes, to benefit other living things. That's what biotechnology is. Living things to help other living things. So genetic engineering, it's a process, all right, that involves identifying one gene. What is a gene? Somebody tell me what a gene is. Okay. What is a gene? It's a piece of DNA, right? A piece of chromosome, right? It codes for one thing, a gene that codes for one thing in an organism, and we're going to transfer it to another organism. So we're looking at one gene in an organism and transferring it to another organism. Usually to give the second organism characteristics it did not originally have. So, if it's something that we didn't originally have, what would the purpose of this be? Why would we do that? Why would we take a piece of a DNA from one organism and give it to another organism to give it a new characteristic? Make it stronger. Ooh, I like that, Christopher. Adapt. Good. I want to give you this word. Enhancement. What does an enhancement mean? More unique. I can guarantee you all know of a genetically modified organism. Every single one of you. Are you ready? Giving you a poll. Have you ever, I'm playing a game of have you ever heard of a labradoodle? A labradoodle. All right, this selective breeding is kind of a way of genetic engineering, right? When we take two dogs that have traits that we want, right, and we mate them to make a new species of an animal, right? Now we could say that's speciation, but that's kind of what we're doing with genetic engineering. It's saying, I like the looks and size of this dog, but I like that this dog is hypoallergenic and has this kind of a temperament, and that is, right? So that's like a selective breeding where I'm picking genes that I want for a dog to do that, right? So that's an enhancement. Now we can do that on a more specific level, and we can do this with recombinant DNA. Everybody point to recombinant DNA and plasmids. I'm not going to make you type it just because it's long, but I want you to maybe even say it out loud at your house. Recombinant DNA. Yes.
Recombinant DNA.
Good. Recombinant DNA. So this right here is a plasmid. All right, we talked about breakfast foods. What breakfast food does a plasmid look like? What does this little plasmid look like? It could be a pancake, you're right. What resembles a pancake with a hole in the middle? Yes! Ooh, a Cheerio too. A bagel. You guys are so much more unique than I was just thinking a donut. All right. Think of a donut right here. These plasmids are circular pieces of DNA. Now, these plasmids are originated in bacteria. All right. So this is a bacterial cell. And plasmids are circular pieces of DNA that we find in bacterial cells. And we use plasmids a lot when we do biotechnology. Why? Because plasmids are they're just easy, right? They're way easier, less complex to work with. So why do we like plasmids? Well, they can replicate independently of the main chromosome. So because bacteria, is it a eukaryote or a prokaryote? There are two types of cells, right, that exist in the whole world. We've got eukaryotic cells and prokaryotic cells. You're right. These are prokaryotic cells. Prokaryotic cells are way more simple. Eukaryotic cells are what me and you are made out of. They're what you are made out of. They're a complex cell. So for us to manipulate DNA, we are going to go and take a prokaryotic cell's DNA. And it's a little different in a prokaryote, all right? They have this main chromosome, but they also have these circular pieces of DNA in their cells called plasmids. And we like them when we are doing when we're manipulating DNA through biotechnology because they replicate independently. That means everything in this whole cell is not going to replicate at the same time. These plasmids are going to be able to replicate on their own. All right. Plasmids also provide genetic advantages because they have antibiotic resistance. What does that mean? Is that they're just strong. Okay. So in chat, I want you to type plasmid. Plasmids equal strong and reproduce alone. Okay? They're super cool. They do their own thing, right? They don't like do what the rest of the DNA in the cell is doing. All right, they're kind of like the go your own way kind of DNA in a bacterial cell, and that's why we love them. All right, they are strong, they are resistant to changes. All right, they are reproducing on their own, and so plasmids are used as vectors. All right, we're going to use this as a bunch of new words today. Vectors in genetic engineering, where other DNA can be integrated into its DNA and then easily replicated. What does that mean? Somebody tell me what that means in their own words. Plasmids are used as vectors in which genetic engineering, where other DNA can be integrated into its DNA. Ooh, Dustin, I love that example. Marla, can you grab me some Play-Doh? I love that example. All right. Marla's grabbing me some Play-Doh. Kinetic sand probably won't work. Our Play-Doh, is it all mixed up?
It's fun.
If you can find a little bit of one color and a little bit of another. We got a Play-Doh ice cream machine truck. And it mixes all of our Play-Doh together. It's super cool.
I remember having a lot of Play-Doh sets at a young age. I still sometimes have them to this day.
Anybody else
They
have?
may have been hidden.
Well, if you have Play-Doh in your house, you'll have to grab some. But this is the cool thing about plasmids is they can easily let's do this. They can easily, so let's say this blue is all the original DNA. They can easily have segments entered, all right, into it. Now I should have made that a circle because they're circular sections, right? But they can easily have pieces of DNA added to their section, and it doesn't affect them. And they just keep going on with life as normal, keep reproducing. And so what does that do? It makes whatever that section that is new, it keeps being added. All right, here we go. All right, so let's make ourselves a cool plasmid. All right, a plasmid is a vector is something that you use to transport something. All right, so yes, Milo, my stage hand is here. So Milo, this is a vector, okay? In the cell, in the bacteria cell, by itself originally, it looks like this. All right, there it is, unscathed. Its normal self, hanging out in its bacteria. And then what happens? We come in and we're like, ooh, I wonder what would happen if you added blah, 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 all right, to this organism. So what we do is we go in, and it does look going to be kind of like this, right? We're going to deconstruct the plasmid, and we're going to add it in there. Now the plasmid looks like this. All right. The original section, this is the original DNA. The blue is the original DNA, purple, I mean, is the original DNA. And the yellow is the new DNA. And now we are able to manipulate DNA into doing something different. All right. So let's look at how that plays out. So this is our recombinant DNA. So here's our plasmid, right? Here's the gene that we wanted to add, and we add it. And then here is our recombinant or recombined plasmid, right? With that gene that we wanted. This is called recombinant DNA. All right. And then I don't even care if in your mind you call it your recombined. Because doesn't that make sense? Recombined DNA. All right, so even if you call it recombined DNA and on your state test it says recombinant, we're like, oh yeah, recombinant. It's really recombined DNA. Okay. Yeah, kind of. So this is recombined DNA where we had the original, we cut it, added a piece, and then this plasmid, this super strong, super reproducible piece of DNA is going to continue on with life. Can I have somebody read this section down here? Just this segment down here.
We have our hands up, so.
Okay, let's have Ireland read this, and Pippo, have you read the questions.
Sure.
Sure.
Okay, wait, just to clarify, I'm reading just the bottom half. Okay.
Yep, if a human.
Okay. If a human gene was added to a bacterial genome, what would the bacteria do? It would create the proteins the human gene codes for while still making the proteins that the bacteria codes for too.
Oh, you're muted.
Thank you. So what that is saying, thank you so much, Ireland, is that if this is the bacterial DNA and human DNA is added to it, that this bacteria, whatever bacteria is lucky enough, right, is going to start producing whatever this codes for in humans. Yeah, Mila made some more recombinant DNA. Say recombinant DNA.
Recombinant DNA.
And what are we going to call it instead of recombinant to help us remember that it's a mix? Instead of recombinant, what could we call it instead? Yep, recombined DNA, right? Recombined DNA. And where do we do it? What is this donut called? Starts with the P. Say
popcorn?
Plasmid.
Plasmid, not popcorn.
A plasmid. A plasmid. Plasmid is a circular little donut piece of DNA in what kind of a cell?
I don't know.
A human cell? A prokaryotic bacterial cell. You guys are rock stars. All right. So DNA fingerprinting is used in a lot of jobs. All right. I had to get fingerprinted to teach, believe it or not. All right. I had to get fingerprinted to do my job as a teacher, but we also use fingerprinting in crime scenes. Why do they fingerprint me? They fingerprint me because they want to make sure that I have a clean background check. That's part of doing a background check, right? They want to make sure I'm not doing anything sketchy and still interfering with your lives, right? That's a mode of protection for all of my students. All right. And so DNA fingerprinting is something they do frequently. If you can think about some of the crime scene cases that maybe you have seen in the past, okay? I know they will go like one time our car was broken into and they went and took around where you put the key in on the car and put a plastic thing on there, and we're trying to pull up any fingerprints that might have been on our car. I've heard some things in the news about, some of the cases that are going on out west in Iowa with those four girls being murdered, or three girls and a dude, or I forget who it was, but they did fingerprinting in that. All right. I'm missing it. 3 girls and 1 dude. I thought that was what it was. All right. They did lots of fingerprinting in that. All right. So what they can find out from our fingerprinting is a lot. All right. So what happens when they do fingerprinting? What is that? Well, let's take a peek. What we do when we get fingerprinting is we can collect DNA. All right. First of all, it's part of your identity, and then we can collect fragments of DNA. And what happens is this is an example of gel electrophoresis. All right. We're learning so many crazy words today. But in gel electrophoresis, what happens is we put a DNA sample here, we put it in a bunch of gel, and then we run an electric current through that gel, and your DNA separates out into different fragments. All right. You can see here, the heaviest are going to move the slowest, and the lightest will move the fastest. So what you're going to do is you may have samples, and then you'll have the original DNA that you're trying to match it up against. Okay. So if we were trying to look at a victim, our victim's DNA might not be number 4. And if we had 3 people that we were thinking that could be possible suspects, we would run their DNA through here, and the DNA samples collected here, and to see like would the victim's DNA have been collected from these suspects' areas of living. Okay. And and if they look like this, we find the match, right? It's just like a game of matching, like when you were younger. If you find the match, then that means that DNA was found somewhere else. It's a really cool process in a not cool situation. All right. What are genetically modified organisms? Moving on to genetically modified of fruits, veggies, organisms. Has anyone talked about like what is organic, what is not organic?
Do you mind if I ask something real quick? I just wanted to bring this up because Ireland asked it. So she was talking about the thing about DNA fingerprinting. So do they like she asked, do they split differently every time, or are they ever the same?
1 second. Perfect. Sorry, they were texting me about Rennie's tumbling class this morning. Can you repeat that question?
Sure thing. So this is like what Ireland asked about. I think it was talking about DNA fingerprinting. So, like she asked, so they split different every time, or are they ever the same?
I need to pop the question up.
Yeah, it's basically the previous slide you shown. If she was asking that question.
Oh, right here. So does this DNA split like this every time? Is that what you're asking? Is that correct? So yes. So like if you ran my DNA through an electrophoresis, every single time it would look like this. It would look the same. There might be like a slight differentiation, like where this one might be a little further back than this piece, but no. So like if if this is a victim and these were 3 suspects in which DNA samples were recovered from different areas, and number 1 we can see matches up with number 4, we'll say, oh my goodness, this victim's DNA was found on victim number 1's car and it matches the victim's DNA. And so that would give it away. So yes, there would be consistency with the as long as the electrophoresis process was ran multiple times. Or, well, what was that?
That scared me for a second.
Yeah. What was that?
The Play-Doh was trying to get you back.
She chucked the Play-Doh at me. You about saw me go down live. The Play-Doh about took me out. Yep, absolutely, Ireland. So just like every single one of our DNAs are different and unique, if every single one of us would run our DNA through the electrophoresis, that would sample out differently. Yes. Absolutely. All right. So if you have seen in the grocery, a lot of times there's going to be 2 types of bananas, 2 types of apples, 2 types of strawberries, raspberries, whatever it might be. And that is going to be your GMO foods and your not GMOs. We call them organic. Our non-GMOs are
I don't know how to
organic.
refresh it. Do you think that'll work?
Oh, somebody need help with their computer? Yep, refreshing it sometimes does help. So GMO means genetically modified organisms. So a lot of the times we will have foods that we change the genetic code in to make it more red, like make your apples more red. They'll actually go in and change the DNA with our little plasmids. You have that Play-Doh, Mila? Bring it on up here. All right. That, yep. Why make an apple more? It's not for the use of anything other than your eyes, right? Like you're going to see it and you want to buy it because it looks like a beautiful red apple. All right. Like in the instance of corn, sometimes they will change the genes of corn to make them resistant to insects. All right, so they have gone through and genetically modified all kinds of food that still grow in our fields or animals that still grow in barns. All right, they do this to not just plants, but they do it to animals and livestock too. They change that DNA to make it more genetically enhanced for the market. Okay, so when you buy a organic product, it is more expensive. Well, why is it more expensive? Well, think about it. Taking care of corn without it being genetically modified for these critters takes more care, right? It's going to take some pesticides, it's going to take some whatever else. And so because they now, is it always worth the price hop? I mean, I think that's debatable, but they are a more whole food, a form of your fruits and veggies and meats that have not been messed with, if that makes sense, genetically and oftentimes with pesticides. Babysitter for GMO. Yes. You mean like the organic versus not? Yes. It tells you. And so now when you purchase those foods, they make you basically identify whatever it is that has been genetically modified from others. All right, really quickly, concerns. All right. What are the concerns about our genetically modified organisms? Well, there are concerns that proteins that are produced from these genes can cause allergic reactions, side effects, and honestly, we don't really all know what it can do because we haven't had a generation that has consumed all these genetically modified like organisms, and there hasn't been a correlated study. All right. But what would happen if genetically engineered plants killed insects that pollinate, like bees and butterflies? What would happen? No. We need our bees, we need our pollinators, we need our butterflies. Right. And if we don't have pollination, we don't have the growth of a lot of those plant species. All right. So by enhancing one organism, we could be endangering or killing off other organisms. All right. What might the impact might be the environmental impact if farmers grew their crops in areas where they normally couldn't? Like, what if we started growing corn in the desert? And we could just grow corn anywhere. That would be weird. It would have a massive impact on farmers because guess what? There would be so many more sources of corn that what would that do to the price of what they were able to sell their corn for? It would tank it. It would make it lower. Maybe I could even grow corn in my yard really easily and I wouldn't even need to buy theirs. Because we made such an easy to grow corn. Right. So these things can be used for good, especially medical. Okay. We talked about how sometimes that can help with insulin. All right. That our recombinant DNA can actually help us make insulin. All right. And that helps our diabetics. So that's a great way to use it as beneficial. There are beneficial reasons. I think that we have to just ethically start being careful and decide when is enough enough? Where is that line? And I think that's what gets fuzzy is where is the line with it's okay to modify this or that, but it's not okay to modify other things. Yes, tumbling is on. I confirmed with her teacher. Sorry. Chris is going to take her into tumbling, and all of our schools are canceled, but tumbling is still on. So what we're going to do is I want you to try this differently today. I want you to pop open your quiz right now. Pop open your quiz in your classroom if you have not taken it. Okay, everybody do that. Let's give you a second. And we're going to answer these together. We actually hit gel electrophoresis pretty well today. I also canceled your gel electrophoresis assignment. So it's just this quiz. Okay? You're welcome. All right. So let's look at number 1. If Pip will read number 1 for me, that would be fantastic.
Sure. Which phrase best defines biotechnology? A, the science that involves using living organisms to produce needed materials. B, the practice of using technology in science experiments. Or C, the use of technology to solve medical problems in living organisms. Thank you. Oh, you're muted.
I'm doing a good job with my microphone today. All right. What do you think, guys? What best defines biotechnology? Remember, bio means living and technology. What do you think? It is a hard one. I took the quiz the first time and I was like, I don't know. It is A. It is A. The science, all right, that involves using living organisms, right? We're going to use our bacteria that is alive and we're going to use it to produce materials, right? Our DNA from our bacteria, we're going to modify that sucker so that it produces something we want it to produce.
Oh yeah, the
In the
answer
case
to
of
Oh,
insulin,
sorry.
this bacteria, if this is the protein to code for insulin, which is something we do, we will take the bacteria, put in the protein, the segment for insulin, and now this bacteria is going to produce insulin that we can use. That was a tough one.
Oh yeah,
All right, what about number 2?
Sorry, just wanted to tell you, the answer choices may be different. They are the same, but the order of them may be different.
Yep,
compared
they
to
are
this.
different orders in your quiz for sure. All right, which tool of biotechnology do investigators use when trying to connect a suspect with a crime? What do you think? Yep. DNA fingerprinting. Good. Sorry, Pip, I just went and read because I'm stressing out about time.
It's all right. Take your time.
And last but not least, you want to go ahead and read this, Pip? That's okay, Tyra.
Oh yeah, I almost clicked on Outlook. I almost, clicked on the thing. But anyways, I will. Which substance will a bacterium produce when a human gene is added to its genome? A. Human carbohydrates. Please eliminate that one. B. A protein made up of both human and bacterial properties. C. Human plasmids that can be isolated from the bacterium. Or D. The human protein coded for by the human gene. Thank you.
What do you think? I feel like this is kind of tricky too, to be honest. I feel like this is a tricky question. I'm going to tell you what I think that you could choose as a second answer. And I want to look and see what you guys are doing. All right. You're all split, right? This is what it is. It is the human protein coded for by the human gene. I personally, at first, I think you could make a case for a lot of these because we talked about plasmids right today. The only thing that's wrong with this one is it's not human plasmids. The plasmid is a bacterial plasmid, right? So it's bacterial plasmids. Human carbohydrates, we don't really produce carbohydrates, right? Carbohydrates give us what? Starts with
Energy.
E. Yes, carbohydrates give us energy, and we get that from our food. So we can eliminate this one. So the one that I think is tricky is a protein made of both human and bacterial properties, because we have this, right? Like, and so much of this plasmid is still bacteria. And then we have this human section. This actually trumps all of this. Crazy, okay? But when you put this into our plasmid, that recombinant DNA, when it's recombined like that, it's going to basically trump those bacterial properties, and it's going to produce that human protein for the gene. Okay. I know that was a tricky question. So just like if you had to say which one is stronger or better, recessive or whatever, the plasmid is going to start producing whatever that new segment that was put in is. All right. So that one would be D. Now, as long as you can tell me what gel electrophoresis is, we're good without taking that quiz. What is a gel electrophoresis? What do we use that for? What is it? It is a tool for looking at what? Good fingerprinting, but not just the fingerprint like its shape, but the DNA, right? Gel electrophoresis is a way of sorting our DNA. Okay? Where the heaviest is at one side and the lightest is at the other, and that everybody's pattern is just a little bit different based on your genetic code. Yes. Good. It's the sorting out of your DNA segments, right? That is what our gel electrophoresis is. It's a tool of sorting and examining DNA. Do you feel good about that? What is recombinant DNA? What's our What's our word for that? We're not going to say recombinant in our class. We say something else. Recombined DNA, right? And I just want you to think of our Play-Doh. Okay, our recombined DNA. All right, why would a scientist insert one DNA of an organism into another one? Why do we do that? That's so weird. Good, to enhance it, to create a change, to make it do something, have a favorable outcome, to create something new, to get something quickly. I love it. Those are all great answers. And lastly, what are some pros of genetic engineering? What do you think? What are some pros? Yeah, it could disenhance. You know what I mean? Like if you wanted to buffer something. What's a pro of genetic engineering? Yeah. I mean, for a farmer who's growing corn, if you didn't have to spray all of your plants with pesticides, they were just resistant on their own. How much money and time are you saving in your fields? I'm thinking that's pretty awesome. In the case of this, where you're producing insulin, my brother-in-law takes insulin shots, right? If we didn't have that remedy for him, his diabetic condition would be a lot worse, right? So we do that with a lot of medical things where we use that recombinant, recombined DNA to produce things that our human body needs and that certain human bodies don't make because of whatever reason. A mutation sometimes, right? But these suckers help us fight some of our human diseases. All right, what are some cons of genetic engineering?
I put it in chat again.
Right, cause allergic reactions. Good. I mean, cons of it are going to be, who knows? Long-term effects are unknown. It could, we always think about how it's favorable for us, but how could it be unfavorable for the insects? How is it unfavorable for our soils? So long-term environmental things. And then I'm going to throw this at you right before we leave. There's also the thing of, how much should you be able to modify humans? Right? They used to say, now that we know all of the DNA in the humans, how come you can't genetically design your own baby? Oh, I like that person's blonde hair and blue eyes, so I'm going to take a piece of that person's DNA and I'm going to stick it in. Right? Starts getting real unethical there. Right now, that's outlawed. But is that possible? Yeah. So sometimes it's like people say it's playing the role of like God or of nature, right? This can be used for a good, but anything that can be used for good can also be used for bad. Yeah. Yep. It's crazy. All right. I'm sorry. I know I'm over. I will stop the recording. I will see you on Monday. It is Wednesday. So if you need me before